Whew. Okay, it's time for chapter 4. Thankfully, I'm getting this up a little faster than I feared because I really want you to have time. However, one thing I do need to tell you is this is not going to be just like if I was doing it in class, because I have embedded videos. And if I play these videos and then post it on YouTube, then it's going to tag me for copyright infringement, and then I can't guarantee you'd be able to see them anyway. So please, 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 please make sure to look at these videos on your own in the PowerPoint called My Chapter 4, which is in the My PowerPoints folder. It's there. Please look at it. I can't stress it enough because one of the videos has this one very catchy part, and it is one of the questions on the test. So you really need to watch it. All right, tour of the cell. Here we go. We are finally to the cell. All righty. We talked about some of this stuff already, and I just realized, wow, my chair is farther back than normal. Our understanding of nature goes hand in hand with the invention and refinement of instruments that extend our senses. So, we talked previously about the microscope, because at least I think we did, or actually we might just do that now. So, we're going to talk all about the microscope. Scratch the fact that I said previously. I forgot. I was thinking that because of me fixing up this PowerPoint. So, we came up with a microscope so we could actually look closer at, cells. We can look at stuff. He compared the structures he saw in the piece of the bark to little rooms, which is cellulae in Latin, and that's where we got the name cell. So all of these tiny little itty bitty things, they're called cells totally because of this dude. Then we have this other guy, I can never do his name right. He actually refined the microscope. He ended up coming up with the different lenses and stuff like that, so then he could actually tell all the different cells. Previously, before this quarter, I used to go much more in depth than this guy and the stuff he did with his microscope and the stuff he studied and whatnot, because I find him fascinating, but I'm trying to keep this closer to our actual textbook and trying hard not to go off on quite the same kind of tangents as I normally do. Clearly, I'm not doing well because I'm already off on one in just the introduction, but I swear, I'm trying. I really am. So, that's our intro. Let's look at the big picture. There you go. Big ideas. Introduction to the cell, nucleus and ribosomes, endomembrane system, energy converting organelles, and the cytoskeleton and cell surfaces. So, without further ado, introduction to the cell. Light microscope. This can display living cells. The greater magnification and the resolution of scanning and transmission electron microscopes can reveal the ultrastructure of cells. Tell you more about this in a second. Magnification. This is the increase in the object's image size compared with the actual size. So when you see like times 100, that means literally that is 100 times what the actual size would be. Resolution's the measure of the clarity of an image. So this is the ability of the instrument to show two nearby objects as separate. I think a storm is coming. Example I give for this in class on a regular basis. I should see what's on the next slide. Okay, it does not show you. Yay. Okay, I used to have this thing and it talked about all the stuff, and that's when I talked about resolution. I think we have one coming later, but I'll cover it now anyway. So resolution, where it says ability of an instrument to show two nearby objects as separate. When you hear that, you're like, of course, but a really good example of this is for anyone who wears glasses. Anyone who has ever gotten glasses in their entire lives, adult lives. I mean, I guess if you got glasses when you were 2 years old, you wouldn't remember this. But anyway, think of anybody out there if you've ever gotten glasses. You go in, you get your new glasses, you walk out. One of the first things all of us do is we go like this. We're like, oh my gosh, and all of a sudden you see individual leaves in the tree. You might have known that there were individual leaves, but until you get those glasses that work perfectly, you don't literally see individual leaves. That is resolution right there. So, modern microscopes, we have the light microscopes and the electron microscopes. Light microscopes, we have phase contrast, reflected light, and fluorescence microscopes. The ones that are normally used in the science lab, when I say the science lab, I'm meaning like college. Those are normally the compound light microscope. So normally you have the two eyepieces you look through, and you have light shining up at the object that you're looking at. Then we have the electron microscopes. These are literally taking pictures of those electrons that we covered in chapter 2. Scanning electron microscope is my favorite because of course I have a favorite microscope. In grad school, I took a scanning electron microscopy class, and I absolutely loved it. It was so fascinating. So these are pictures I took when I did the scanning electron microscopy class. So these are images of a fry, a baby, an itty bitty fish before it even hatched. These were the fish that I studied. I studied a fish called the Cryptolevius marmoratus, mangrove killifish. It is one of the few truly internal fertilizing hermaphrodites. So what it does is it actually fertilizes its own egg inside its body before it drops the egg. Terribly fascinating. Love these little fish. This guy, so teeny tiny. And so these are images I took. So this is obviously of the fish, and I want my laser pointer. Earlier I was using green. Let's see if it's still green. Nope, it's red. I'm going to change it to green because I like green. There we go. So here's my fish. This image right over here, and by the way, notice it tells you times 3,500. So massively magnified. This image right here is in here. Cool, right? And then if you look at this pectoral fin right here, this is zoomed in closer. So notice times 370. And then this is times 950. So this right here is this right there. And so this gets to show you just how close and detailed you can get with the scanning electron microscope. Pretty cool. Okay, so in the 1800s, early studies of cells led to what's now known as the cell theory. This states that all living things are composed of cells and that all cells come from other cells. That means they don't just spontaneously appear. They had to come from somewhere. So we have a checkpoint question because of course we do. We've been, on this PowerPoint for 4 slides. We have to have a checkpoint. Anyway, which type of microscope would you use to study A, the changes in shape of a living human white blood cell, B, the finest details of surface texture of a human hair, and C, the detailed structure of an organelle in a liver cell? Well, here you go. You would use a light microscope for the living human white blood cell. Then, for the finest details, you would use the scanning electron microscope like I just showed you with the fish. That shows you just how detailed you can get that texture because that's what I was showing you. And then for the detailed structure of an organelle in a liver cell would be the transmission electron microscope. The reason you cannot use the electron microscope for anything living is because of the process you have to go through to put it in the microscope. It's not a microscope like you would ever think. And if it interests you, shoot me an email. I'll tell you all about it. Or ask me in class because we meet next week. Yay! Okay, anyway. Moving on. So here's variations of these pictures. We have a paramecium. So we have the light micrograph. So that would be your typical type of microscope. Then we have the scanning electron one. This guy, not alive anymore, but look at the detail on the surface. Pretty cool. And then, yes, it's colorized because the scanning electron microscope does not have any color. You have to add that in for the pretty picture. And then differential interference contrast micrograph. So here's where they actually added some stuff to make stuff pop out in the image. Notice this is 230 times, this one's 380 times, this one is 580 times for the sizes. This is the part about the resolution. Unaided eye. You can typically see, say, a human all the way down to the paramecium if you needed to without a microscope. Now, okay, seriously, kiddies? Now, you can get down to that, but a lot of people aren't going to see much better than the frog egg. So right around here is normally where you start using the microscope. So here we go. You start using the microscope right around the frog egg. And you can use the light microscope all the way down here to bacteria. So most bacteria right here. You can use the smallest bacteria is just outside the range because the light microscope is going to have its limits. But most of the things the average person is going to look at is right here. Then you get the electron microscope. Notice this is where you have viruses. So anybody studying the coronavirus right now are probably needing to put it through the electron microscope for the really cool pictures. Alrighty. Microscopic size of most cells provides a large surface to volume ratio. The plasma membrane, which is what's on the outer side, phospholipid bilayer with embedded proteins. We talked last time about the phospholipid bilayer. If you remember, hydrophilic heads, hydrophobic tails. Some proteins will form channels that will shield ions and other hydrophilic molecules as they pass through the hydrophobic center of the membrane. Again, has to be shielded because hydrophobic, they don't want anything wet touching them, they're scared. Other proteins will serve as pumps using energy to actively transport the molecules into or out of the cell. Microscopic size of most cells provides a large surface to volume ratio, which is what I already said. And it's so funny because as I was as I was clicking, I was sitting there trying to decide or remember whether or not we cover the fluid mosaic model in here. And if not, I need to make sure to talk about it. Because the fluid mosaic model is just, I like to make sure you understand what mosaic means, which means it's all made up of a lot of different parts. And that's, a lot of times they use the phrase fluid mosaic for the way the cell works in the membrane. Anyway, here is the surface to volume ratio. To convince yourself that a small cell has a greater surface area relative to volume than a large cell, compare the surface to volume ratios of the large cube and one of the small cubes. So here they're showing you two different cubes. One is one entire unit and one is a bunch of little ones put together. So this is what they're doing. It's math. I don't normally make everybody do this because I find it interesting, but I have discovered most students cannot stand doing this part and they get overwhelmed. Basic takeaway here is surface area to volume ratio is smaller for the large cube because it has more volume inside, less surface area. When you look at the smaller cubes, there's less volume on the inside, more surface area, which is the outer part. Excuse me. I normally end up talking about this with ground beef and steak. Sometimes I talk about this in the environmental science class when I talk about meat glue and stuff. And then in this class, sometimes I talk about it when we were covering proteins, and then bacterias. Other times I mention it later. So basically think of these two things like this one right here is your steak, and this one right here is your ground beef. This one, the surface area, everything's on the outside. All this stuff on the inside can't be touched. This is why you can get a steak that is rare, because you have cooked the outside. The inside hasn't been touched. But this one, because it's all ground up, you have all these little pockets. All this surface area is where the bacteria can build. And so this would be like a hamburger patty. So you have all these little bits of meat made up into one patty, but you have all these little spaces in there. So, yeah. Does that help you with the ratio? No. Well aware of that. But it's one of the things I normally talk about, because I like you to know that if you're ordering a burger somewhere, you should not be getting it rare or even medium rare, because the amount of bacteria that might be proliferating inside. So there you go. All right, plasma membrane. Here is the fluid mosaic model. We have different types of ways to get the stuff from outside to the inside of the cell, vice versa. This is showing you your phospholipid bilayer. This is the phospholipid. This is a phospholipid. Bilayer means two layers. So hydrophilic heads on the outside, hydrophobic tails on the inside. All cells have a plasma membrane, DNA, ribosomes, and cytosol. Bacteria and archaea are prokaryotic cells. Prokaryote means there's no nucleus. All other forms of life have eukaryotic cells. EU is true. So a eukaryote is a true nucleus. So all of these cells will have a nucleus. Eukaryotic cells are distinguished by having a membrane-enclosed nucleus and many membrane-enclosed organelles that perform special functions. So, prokaryotic cells are smaller and simpler in structure. These are a lot more basic. They are lower on the evolutionary scale, which we will cover in Bio 124. And when we're in 124, you're going to hear me constantly refer back to 123. So here we go. This is a typical rod-shaped bacterium. There is no nucleus. Instead, we have a nucleoid. So they have most of the same material in there, but it's kind of like blue instead of perfectly contained. We have the cell wall, the capsule. We have the plasma membrane holding everything inside, and we even have these flagella, which is how they move around. Membrane-enclosed organelles compartmentalize the cell's activities. So the organelles are like their own tiny little cell organs, basically. So it's the little specialized pieces inside the cell. And one of the videos that I want you to watch that I can't play on this one is an entire song, and they keep going on and on saying cells, cells, they're made of organelles. That's what this is talking about. So, nucleus and ribosomes carry out the genetic control of the cell. Organelles involved in the manufacture, distribution, and breakdown of molecules include the endoplasmic reticulum, and you will often see it just as ER. The Golgi apparatus, which is like UPS, you box it up and ship it out. Lysosomes, which is like the recycling center. So they take the stuff and go, oh, we're not going to use this. They break it down and reuse it or chuck it. The vacuoles and the peroxisomes. Mitochondria, the powerhouse of the cell. Everybody should have learned that at some point in school, whether it's junior high or high school. Mitochondria, powerhouse of the cell. It's because that's the one making the energy. That's what it means by the powerhouse. And structural support, movement, and communication between cells and the functions of the cytoskeleton, plasma membrane, and plant cell wall. So that's all these different parts. Now, checkpoint question. Identify the structures in the plant cell that are not present in the animal cell. Well, plant cell has Chloroplasts, a large central vacuole, cell wall, and plasmodesmata. I normally don't ever ask you about the last part, but knowing that the plant cell has a cell wall and the animal cell does not, that's very important to remember. Central vacuole, that's an important one because that's kind of what gives the structure to the plant. We talk about turgor. We talk about that later, and that's due to the central vacuole. And then the chloroplast, that's what makes it where they can conduct photosynthesis, which of course you will need to know later. So here is a generalized animal cell. Just when you look at it. If you go online, if you have done the cell quiz, you've already seen the image I will use for labeling the cell on your test. If you have not yet done the cell quiz, what are you waiting for? You should totally do the cell quiz because it has the image I'm using on the test. The reason I do this is because each one of these images make it look a little bit different and it makes it a little harder. They're all still exactly the same things, but it gets a little difficult to identify some of the stuff. But look, here is the powerhouse of the cell, the one that looks almost like a cut open jelly bean. Okay, this is the plant cell. Notice we've got the plant wall, we've got the chloroplast, we have the important stuff here. All right, cells, they're made of organelles. So if you are doing one of these, oh, I guess I can't do it with the laser pointer. Will it do it without the laser pointer? Yeah, there we go. If you do this, you would just press right there and then you get the video. You listen to it. They're going to sound really stupid, really annoying, really juvenile, but I promise you, if you actually listen to these even once or twice, you will remember most of your cell and you will remember what the cell parts do. So this is one of the best things you could do for yourself to actually learn. So I have that one, then I have this one, and then this one is really bad. Oh my gosh, I think this one's all about the lysosome. You'll remember that it digests and recycles. I'd love to know if any of you actually watch this and tell me what song it parodied. It's kind of funny. And then this one is a serious student one. And I say serious student one because this is one of those, and now we are going to talk about the cell kind of videos. But it has really good information. So all of these are embedded. That means if you download my PowerPoint, my chapter 4, and then play it in slideshow mode, you can then just hit play. If you don't put it in slideshow mode, then if you right click, you can still do play the video. So seriously, watch the videos. It will help you tremendously. And one of the questions on the test comes from the videos. So now let's talk about some parts of the cell. We have the nucleus and ribosomes. Nucleus houses the cell's DNA. So this is what holds all of that. And then that makes the messenger RNA, so then it continues to make the proteins, blah, blah, blah. So subunits of ribosomes are assembled in the nucleolus. Now, checkpoint question, describe the processes that occur in the nucleus. Well, the DNA is copied and passed on to daughter cells, which you don't know about yet because we haven't covered this yet. rRNA is made with ribosomal subunits assembled. Protein-making instructions in DNA are transcribed into mRNA. We have only barely touched on that. I have an issue with the layout of this so far, but again, I'm trying to just go with the flow, go with our new textbook. I think we should cover all that before they try to do a checkpoint question. But anyway, ribosomes. These are composed of ribosomal RNA and proteins. They synthesize, which means they make proteins according to the directions from the DNA. Cells that make a lot of proteins have a large number of ribosomes because the ribosomes are what's helping make the protein. And that brings us to the endomembrane system, because when we talk about the endoplasmic reticulum, we have to have heard about ribosomes first. So that was a good order. Now, many of the membranes within the eukaryotic cell are part of the endomembrane system. So many of these organelles interact in the synthesis, distribution, storage, and export of the molecules. We have the endoplasmic reticulum, which I told you you might just see listed as ER. We have the smooth ER and the rough ER. The smooth ER synthesizes lipids and processes toxins. The rough ER is where we get our ribosomes. So if you can just remember R for rough and R for ribosomes, you will be able to remember that the rough ER has the ribosomes. And when we see the image, you're going to see that it blends from the rough ER to the smooth ER and then goes out. So explain why we say that the endoplasmic reticulum is a biosynthetic workshop. Well, because it's producing a whole lot of stuff. So we are making phospholipids for cell membranes, steroid hormones, and proteins for membranes, and then other organelles and secretion by the cell. So the endoplasmic reticulum, well, I was about to say is key, but quite frankly, everything in the cell is key. So just, scratch that. Now, the Golgi apparatus, a moment ago, I referred to this as UPS. This one consists of stacks of sacs. Oh, that's like a tongue twister there, isn't it? Stacks of sacs, in which products of the endoplasmic reticulum are processed and then sent to other organelles or to the cell surface. I am so tired of sitting up. This is my third video today, and I am getting very, very, very fidgety. All right, so here we go. What happens here is it's going to go from the Golgi apparatus, and then it is going to move on into the ER. So here it's just showing you the receiving side of the Golgi, then it goes through, and then it transports, and then it ships it. That's why, again, the Golgi apparatus, just think of it as UPS. It takes it in, packages it, ships it out. So what is the relationship of the Golgi apparatus to the endoplasmic reticulum in a protein-secreting cell? Well, right there. The Golgi receives transport vesicles budded from the ER that contain protein synthesized by bound ribosomes. The Golgi finishes processing the proteins and dispatches the transport vesicles to the plasma membrane where the proteins are secreted. So, again, it takes the stuff, packages it up, and ships it out. So, seriously, if you can just remember that Golgi is like UPS, you got it. Now, lysosomes. Here is a very fascinating, well, you'll see. I add stuff to here that's not in your book. So lysosomes house enzymes that break down ingested substances and damaged organelles. Earlier, I made a reference to them being like recycle bins and trash cans. And I said recycle bins, and then I said digest and recycle. And that's in one of the videos that I suggest you watch. So, how is a lysosome like a recycling center? Because it breaks down the damaged organelles and recycles their molecules. So this is where it's like, all right, can't use this, and then they use it all up. So here we go. We have the digestive enzymes, the lysosome, and the plasma membrane. So something's coming in, and we've got this food vacuole. Then the digestive lysosome goes, oh, hey, we could use this. Let's break it down. And then it digests it, so digests and recycles. So now the vesicle containing the damaged mitochondria, and so this can't continue, the lysosome comes over here, gulps it up, digests it, and ships it out. So I shouldn't say ships it out, I should say disperse, because recycle. Now, this is one of the parts that I'm adding in here is stuff that can happen when there's a lysosome malfunction. When a lysosome does not work properly, some of the cellular material is not properly recycled. So that means something might go wrong. Imagine, if you will, your garbage can never getting changed. So if the lysosome is not taking it out, it just keeps piling up. So that's one of the ones I'm going to talk about here. That brings us to something called Tay Sachs disease. Tay Sachs disease, genetic mutation that alters the lysosomal enzyme that breaks down gangliosides, which accumulate in nerve cells. These kids who have this typically die before the age of 5. What happens is it is a fatal genetic lipid storage disorder. And so by that lysosome messing up, these fatty substances that typically would not have been an issue because the lysosome would just break it down and, redistribute. Instead of that happening, it just keeps building up and building up and building up. So this keeps building up on tissues and nerve cells in the brain. And then, it breaks it all down. So a baby with Tay Sachs disease will appear to develop normally for the first few months. Then, as the fatty substances that should have dissolved don't, and they're getting built up, it begins to slow down and shut down different sections of the brain bit by bit by bit. The child eventually becomes blind and then deaf and then can't even swallow. So clearly the poor baby's going to go. So a person with Tay Sachs, a baby with Tay Sachs disease, is known to have cherry red spots in their eyes. This is kind of a way that they see it. Well, they notice that the baby's not developing properly, and then that's one of the indicators. You see those spots. Incidence of Tay Sachs is particularly high among people of Eastern European and Ashkenazi Jewish descent. Patients and carriers of Tay Sachs disease can be identified with a simple blood test. So they just check it in order to see if both parents have to carry this gene in order to have an affected child. When we get farther in this quarter and we cover genetics and we do the Punnett square, you will see that it's basically a 25% chance of having a baby with Tay Sachs, but both parents have to carry the gene. If only one carries it, you're safe because it is a recessive gene. No known cure. They don't know how to make the lysosome suddenly start working. And so here, this one said that most of them die by the age of 4. So 5 is reaching pretty far. Okay, so vacuoles function in the general maintenance of the cell. Vacuoles are large vesicles that have a variety of functions. Some protists have contractile vacuoles. Plant cells, which we already mentioned, have the big central vacuole, and that will store molecules and wastes and help facilitate growth. So, checkpoint question. Is a food vacuole part of the endomembrane system? Is it? Yes. It forms by pinching in from the plasma membrane, which is part of the endomembrane system. So, membrane. That's the way to remember it. Organelles of the endomembrane system are interconnected structurally and functionally. This is showing you how it works. So we have our Golgi apparatus. We've got the membrane here. So this begins the whole thing. So something's on the membrane. It goes in here, goes through the Golgi. So remember, UPS. It accepts the package, it processes it, and it ships it out. As it ships it out, here it's showing you go to the lysosome. And here we have our rough and our smooth ER all connected. All these things are together. So the rough ER have the little dots. Those are our ribosomes. And the smooth ER does not have the dots because there's no ribosomes. That's more for the lipids and stuff. So how do they tie together? Well, the transport vesicles move membranes and the substances they enclose between components of the endomembrane system. So there you go. Peroxisomes are metabolic compartments that do not originate from the endomembrane system. So these are separate. Energy converting organelles. Of course, we have to talk about the mitochondria because it's the powerhouse of the cell. Mitochondria have two internal compartments. The intermembrane space is the narrow region between the inner and outer membranes, and the mitochondrial matrix contains the mitochondrial DNA, ribosomes, and many enzymes that catalyze some of the reactions of cellular respiration. Catalyze just simply means to speed up. So, what is cellular respiration? And trust me, we're totally covering this in a future chapter ad nauseum. Cellular respiration is a process that converts the chemical energy of food molecules to the chemical energy of ATP. Now, let me tell you, by the end of this quarter, in person, when I say ATP, I will stop for a moment and expect you to respond with energy. So if I all of a sudden say, so ATP, I expect you to say energy. And then if I say in class, so what is ATP? I expect your answer to be energy because, yeah, we will cover it a lot. So photosynthesis is the conversion of light energy from the sun to chemical energy of sugar molecules. And oh yeah, you'll be learning this chemical equation right here. Chloroplasts are the photosynthesizing organelles of plants and algae. That is why the plant cell has chloroplasts, but the animal cell does not. So, checkpoint question. Which membrane in a chloroplast appears to be the most extensive? And why do you think this might be the case? Well, the thicolloids are the most extensive. That's these guys, the big folded up things. And it's because the chlorophyll molecules that trap solar energy are embedded in there. So if we need a whole bunch of chlorophyll in order to conduct our photosynthesis, then the thing that's holding all of those should probably be the most extensive. Endosymbiont theory. So this states that the mitochondria and chloroplasts were formerly small prokaryotes that began living within larger cells. Basically, it's suggesting that it absorbed it and brought it in. They worked together, and that's what evolutionary-wise led to this. So the checkpoint question. All eukaryotes have mitochondria, but not all have chloroplasts. So what is the evolutionary explanation of this? First, endosymbiosis would have given rise to eukaryotic cells containing mitochondria. Okay. And then there was a second one that gave rise to the cells containing chloroplasts as well as mitochondria. So it diverged and then diverged. Cytoskeleton and cell structure. Cytoskeleton includes the microfilaments, the intermediate filaments, and the microtubules. Functions include maintenance of cell shape, anchorage and movement of organelles, amoeboid movement, and muscle contraction. Checkpoint question. Which component of the cytoskeleton is most important in, and I can't believe I forgot to erase that 2 of 2, holding the nucleus in place within an animal cell, guiding transport vesicles from the Golgi to the plasma membrane, and contracting muscle cells? Well, here you go. Intermediate filaments for the holding the nucleus in place. Guiding the transport vesicles would be the microtubules, and contracting muscle cells will be the microfilaments. I don't have a great handy thing to tell you for this one. Nope, I don't have a good thing. All I keep thinking here is the tube and the Golgi, so I'm thinking UPS. I like it when it does that. So I'm thinking UPS and the subway, but I don't think that's really going to help anybody. All right, so in the 1940s, biochemists first isolated and identified the proteins actin and myosin from muscle cells. Then in 1954, scientists used the newly developed techniques of microscopy because they had better microscopes here to establish how the filaments of the actin and myosin would interact in the muscle contraction. And then in the next decade, 60s, researchers identified the actin filaments in all types of cells. Then in the 70s, they were able to visualize actin filaments using the fluorescent tags in the living cells. And then in the 80s, biologists specifically were able to record the changing architecture of the cytoskeleton. So this is an example of how science and scientists are continuously looking back at the same thing and gaining more and more knowledge. This is also part of why with the current issue with coronavirus, you hear one thing and then a little bit later it goes a little farther and then a little bit farther. It's because that is how science works. You learn all you can at this stage, and then you expand how you can study it. Because if you start here, you're never going to know all of it. You have to start small and then keep expanding your knowledge and keep learning. And sometimes you will learn that something's a little different than you thought before. It's not that, oh my gosh, they just keep saying things. It's a process. You have to learn so you can learn more. Nobody can know everything at once. It's kind of like you had to learn how to add 1 + 1 before you could learn to multiply, before you could then look for a variable. So you have to start at one spot. I guess I should say you had to learn how to roll over and then get up onto your hands and knees for crawling before you could get up and walk, and then you could run. That would probably be better, because a lot of people would say, I just don't like math, but I'm pretty sure most of you at least sit up and presumably walk. So, alrighty. Eukaryotic cilia and flagella are locomotor appendages. It's how you move. It's how these cells work. It says the microtubules are in a 9+2 arrangement. I think we're going to have pictures of this, but I really don't ask you about this. Flagella are longer than cilia. Cilia look almost like little hair-like structures. Flagella looks more like a whip. We had a picture a moment ago with that capsule bacteria. Cilia work more like the oars of a boat. And the flagella can be like this, or it can be like that. Although differences exist, flagella and cilia have a common structure and the mechanism of movement. So they're both just working on movement. So, checkpoint question. Primary ciliary dyskinesia, PCD, also known as immotile cilia syndrome, is a fairly rare disease in which cilia and flagella are lacking motor proteins. This is characterized by recurrent respiratory tract infections and immotile sperm. How would you explain these seemingly unrelated symptoms? So how would you compare those two? Well, without motor proteins, the microtubules cannot bend, so the cilia cannot clean the respiratory tract because your lungs are full of cilia. That's the little hair-like structures. I cover it more in Bio 124 when we talk about it, and when I go off about why smoking anything at all is not a good thing, because you're literally burning the little cilia, the hair-like structures in your lungs. And then the sperm cannot swim because the flagella, that's how the sperm move, so it would kill the motility. Animal cells synthesize and secrete an elaborate extracellular matrix, ECM, and this is what binds the tissue cells together, supports the plasma membrane, and communicates with the cytoskeleton. The ECM might also attach to the cell through other glycoproteins and then bind to membrane proteins called integrins. Here's a picture trying to show you. You see the extracellular fluid, and it's trying to show you how it all blends together. So, according to this image, describe the structure that provides support to the plasma membrane. Can you do it? Membranes attach through membrane proteins to microfilaments of the cytoskeleton and to connecting glycoproteins and collagen fibers of the ECM. So it's just trying to show you would have to look at all this in order to know. And I realize I turned off my laser pointer. Oops. Adjacent cells adhere, interact, and communicate through specialized junctions between them. This is one of those few times in science where the words are exactly what they sound like. So, tight junctions provide cells or bind cells to form leak-proof sheets. So they're tight to keep it from leaking. Anchoring junctions rivet cells into strong tissue, so anchors. And gap junctions allow the ions and small molecules to flow from cell to cell, so they have a small gap. There you go. So, checkpoint question. A muscular tear injury would probably involve the rupture of what type of cell junction? So, which of these do you think would mess it up if you tore a muscle? Anchoring junction. Because right here, rivet cells into strong tissues. That's why. This is just trying to give you ideas. We've got our anchors, we've got our gaps, we've got our tight. So there you go. Plant cell, but not an animal cell, has a rigid cell wall. Because again, animal cell does not have a cell wall. Protects and provides skeletal support that helps keep the plant upright and is primarily composed of cellulose. That was me thinking. I think we covered this we did. We covered this in chapter 3. Okay. I talked about the collard greens and everything. Plant cells have cell junctions called plasmodesmata. I was flipping my syllables somehow. This allows the plant tissues to share water, nourishment, and chemical messages. So, which animal cell junction is analogous to plasmodesmata? See, it's trying to make you think of words together. This would be a gap junction, so it lets it go back and forth. Eukaryotic cell structures can be grouped on the basis of 4 functions. We have genetic control, manufacturing, distribution, and breakdown of materials, energy processing, and structural support, movement, and intercellular communication. Checkpoint question. How do mitochondria, smooth ER, and the cytoskeleton all contribute to the contraction of a muscle cell? Well, the mitochondria gives the energy in the form of ATP because mitochondria is the powerhouse of the cell. Smooth ER helps regulate the contraction by the uptake and release of calcium ions, and the microfilaments function in the actual contractile apparatus. So filaments, they're pulling, so that's what that would be. Handy little table, lots of information. If you want to print out just the table, open up the PowerPoint, right click, and then you can get just the image. So it puts it all together in a nice, simple little area. Now, you should be able to do a whole bunch of different little things. We will go more into other stuff about the cells next week. However, for this one, we have at least finished this chapter. And this is a handy thing to take a look at. In the quiz, it's going to ask you some stuff about metric measurement. I have previously done labs on conversion because the US is pretty much one of the only places that has the crazy comparisons instead of just using the metric system. So science is going to be the metric system. Most places are going to be the metric system because it's actually a very easy conversion. If you look, and I need to get my little laser pointer. If you look here, 1 meter to 1 centimeter, we just go down. So if you have 1 centimeter, notice this is 10 negative 2, you just go, I should go there first, right? Yeah. Okay. You're going to just go over a little bit. So over 2. So this is 1/100th because 2. 1/100th of a meter. This is 1/1000th of a meter. So -2, -3. And you just keep on going down that way. Just move your decimal over and count. That's literally it. Because if it is 1 meter, you go Yeah, 1 meter, you go over 2 zeros, that gives us 100 centimeters. Or you go the other direction, so 1 centimeter, you would take this decimal and go 1, 2, which is why you have 1, 2. You put the decimal over this way, 2 spaces. Millimeter, 1, 2, 3. Micrometer, since it's -6, you go 1, 2, 3, 4, 5, 6. And that's honestly all you do. So it's actually very easy conversions. However, if you try to do it our way, 1 meter is 39.4 inches, but 1 centimeter is 0.4 inches, and you can't even figure the rest of these out. So the metric system, extremely helpful. You should probably check into it. This is the end of the slide. So yes, of course, I'm putting it on there, just reminding you, cell, plant cell, animal cell. And I want to remind you, this is the end of week 2. I started to say week 4, it's chapter 4. End of week 2. Make sure to actually give me detail, actually answer the questions that I tell you to answer me about for the hybrid assignment. And please, please, please don't forget that the first exam is the end of this week. It's an online exam. You will have taken it before we ever even meet, but we meet next week. Yay. We have a real in-person lab next week. Yay, yay. Review your quizzes. Look at your quizzes. 100% look at your quizzes. If you did the extras, you're in even better shape. Think about some of the random stuff I told you in these video lectures that are a good thing to remember. That would be a good thing to remember for your test. And 100% make sure to have done that cell quiz multiple times before you do your test. Your test, you get one chance only. That's it. So that's it. I'm done. Caught up. Everything up for this week. Yes. And random dogs barking. So that's my cue to be done. All right. Peace out.